Hello, good afternoon. We will start in 2 minutes. Hello, good afternoon. Yeah, we are ready, but we will start in 2 minutes. As usual, we know the story and we know that some people can arrive at the last moment. So yeah, we will start very, very soon. Hello, good afternoon. In 1 minute, we will start. Yeah. Okay, so now we can start. Hello, good afternoon all, and I hope you are fine and ready for this Tozo Bioscience webinar. So I'm Didier Sampere, working in the Avantor organization in the global marketing team to push and promote chemicals and chromatography products. Today we will present a webinar called Novel Approaches to Antibody-drug conjugate analysis. Andrea Krum from Tozo Bioscience will talk about antibody-drug conjugates and their characterization. We know all that these cancer biotherapeutics are crucial to fight this serious illness. So, with this webinar, Andrea will present you some recent UHPLC methods, and I'm personally convinced that after this presentation, you will be more comfortable to separate such difficult substances. And before moving to this presentation, feel free to ask some questions using the questions box, and we will try to answer later. This webinar will be recorded, so I think that it's important to you to know that, because after you can share this webinar with your colleagues. And now I think that we can wish to Andrea a great presentation. So Andrea, up to you to start.
Thanks a lot, Didier, for that kind introduction. I would also like to welcome everyone in the audience. I hope you're equipped with a tea or coffee and you want to learn a couple of new things now. The topic we will cover today is the analysis of antibody-drug conjugates. I'm quite aware you know what it is, otherwise you probably would not listen to us now. Still, I would like to start at the very beginning of antibody-drug conjugates, because quite interestingly, it's not something that just popped up recently, but first ideas around the magic bullet, for instance, were already out there in the very early 20th century from Paul Ehrlich. However, it took some time until the first antibody-drug conjugate was actually produced. And fair enough, the first ones actually were not successful in the clinics. These have been marine antibodies that were associated side effects. However, with the beginning of 2000, the first antibody-drug conjugates were approved by the authorities, either FDA or EMA, and up to now we have 11 antibody-drug conjugates in the clinics already that were approved, and there are a lot more than 100, which are currently in clinical trials. Now, why do antibody-drug conjugates actually get such a success now? I think, and I hope you all agree, that the concept of an antibody-drug conjugate, or short ADC, is actually a quite clever one, because what we basically have is our antibody, which, like all other antibodies, also target a specific protein, which can be, for instance, on a cell that we don't want to have in the body. May it be a tumor cell, for instance. So we have this protein structure which specifically binds to our, let's say, tumor cell, for instance, and this one is linked to a cytotoxic drug. The cytotoxic drug can then act in the cell where it's specifically targeted to induce cell death in this specific cell. Apart from these two very critical components of the ADC, what we also find is a linker, so the molecule which links, as the name says, our antibody to the chemotherapeutic drug. And this is typically cleavable, because what happens if, like, the whole ADC is applied to a patient, the antibody will go to the site of action, bind there, the whole molecule will be internalized by the cell, and only then the linker will be cleaved in order to release the cytotoxic drug, which can then induce cell death in the cell of interest. Now, this is the very basic concept, but what we see recently is that there are some trends in the development of antibody-drug conjugates. One of the routes that are taken at the moment is to try to get site-specific conjugations. What that means is we just attach our chemotherapeutic drug to a known site or a known place in the antibody structure. So this is something that can be done, for instance, by just using cysteine for coupling the chemotherapeutic drug, or glutamine, for instance, or even artificial amino acids that we introduce into the sequence of the antibody, and we then use it to link the linker plus payload onto the antibody. So what we will get is defined drug-antibody ratios. Why is that important? Because you can imagine the number of drugs which is attached to our antibody has a huge impact on how efficient it is and how safe the molecule is at the end. And if we can control the number of drugs on the antibody, that's of course an advantage. Secondly, not only full antibodies are currently used in order to attach the chemotherapeutic drug onto the molecule, but what can also be used is not the whole antibody, but a fragment thereof, which still has the specific binding activity, but it's of course a smaller molecule in general. This, again, you would not do it if it wouldn't have any advantage. It can better penetrate solid tumors, and also the internalization of the whole molecule into the cell works better. However, there's also a drawback, which is shown here basically. We do have better penetration and internalization. However, we also have a shorter half-life of those molecules, which is smaller. In order to overcome these drawbacks, what we could use is, for instance, polyethylene glycol modification, of those molecules, which make them more hydrophilic, also even increase the size a little again, but that leads to an elongation of the half-life, or to use hydrophilic linkers, again making the molecule more hydrophilic and increasing the half-life of the molecule. Now, we know already quite a lot about ADCs. As I showed you all the structure in the previous pages. And this actually also explains why analysis of ADCs is such a challenge. So if we just think of a monoclonal antibody as one part of the ADC, it already comes with a high complexity itself. We may have oxidations on the molecule, reductions on the molecule, different glycan patterns on that molecule, all making it complex itself. And on top of that, we have our chemotherapeutic drug. First of all, that we need to know if it's the right drug without any modifications. And of course, we need to know about how these both molecules are put into relation. So where is my drug attached to? How many of the drugs do I have attached to the antibody? And all of this gives quite a complexity to look at already, as compared to just having one of those as a therapeutic entity, for instance. Now, another additional challenge, I would say, to the analysis of ADCs is that due to the chemotherapeutic part, which typically is a hydrophobic modification of the molecule, makes the whole molecule more hydrophobic. On the one hand, this leads to a higher aggregation propensity. And secondly, we may have more issues with hydrophobic interactions with stationary phases that we want to use in our HPLC or UHPLC analysis. So even though there are a lot of things that can be analyzed on an ADC, today, due to the time that we have, I will mainly focus on the analysis of intact antibody-drug conjugates and not go too much into detail into analyzing one molecule, so either mAb or drug separately. First of all, even though the name for today's lecture is actually novel approaches, I would still like to start with something very conventional that is used to analyze ADCs. So most of you are probably aware of using hydrophobic interaction chromatography, or short HIC, to analyze the drug-antibody ratio on ADCs. Our drug-antibody ratio, I think it's self-explaining, is the number of drugs on average that we find on our monoclonal antibody in an antibody-drug conjugate sample. Now, as it impacts on, I said it before, the efficacy, but also the safety of the whole drug sample, it's an important characteristic that needs to be determined in a sample. Now, one of the gold standard methods is to use hydrophobic interaction chromatography, because we heard it before, the chemotherapeutic part makes the molecule more hydrophobic. So we can use HIC to separate our sample components by hydrophobicity in order to get back to how many drugs do we have attached onto the antibody. Now, what we see in the first chromatogram here is how the antibody without any conjugation looks like when it's analyzed by HIC. What we see is one main peak that eludes around 9 or 10 minutes. And now if we look at an antibody, also trastuzumab, that is conjugated to hydrophobic chemotherapeutic drugs, what we will see is, again, a portion of the sample which elutes at the same time as the molecule without conjugation, meaning this is actually without conjugation, so drug-antibody ratio of 0. Now, what we will find in addition is molecules or sample components that elute later, meaning they are more hydrophobic and they have more drugs attached to the antibody. And this way you can determine how many drugs are on average attached to your antibody molecule to determine the drug-antibody ratio. And this works by looking at the peak areas basically and counting them together and calculating the average DAR. Now, just a very short reminder on how hydrophobic interaction chromatography works. Why can we separate the hydrophobicity? So what we do is we inject our sample, the antibody-drug-conjugate sample, in a very high salt concentration buffer onto our stationary phase. Stationary phase, which is our HPLC, UHPLC column, has also a hydrophobic surface. Now, if we have a lot of salt ions, then we don't have a lot of solvation of our molecules. So everything that is hydrophobic on our molecules will be accessible and can interact with a hydrophobic stationary phase, meaning we have binding. Interaction is binding. Now, in order to elute our samples again, what we do is we decrease the salt concentration so that we get back the solvation of our molecules. We get a rigid water layer on those, and we don't have access of the hydrophobic parts of the molecule anymore, and they will elute. So in short, we have high salt concentration when applying our sample, and for elution, we reduce our salt concentration. Now you probably think like, yeah, what's the novelty of it? And one of the things that we as TOZO thought about is there a possibility to get a higher separation performance and a higher reproducibility? So we developed a new Higg column. It's actually called Higg ADC butyl column. The butyl is the butyl ligand that is attached to make the stationary phase hydrophobic. And what we mainly focused on for development of this column is really the repeatability, meaning stability from column to column, from injection to injection, lot to lot, and so on. And the separation performance, which we mainly achieved by allowing for homogeneous particles and a good packing quality. And this is something that we can see here. I don't not only want to talk about it, I also want to show you that we actually get a very good separation performance with this new column. And so what we did is we compared the new column to a couple of other columns on the market. And what we see is actually that we have a very good retention, but also again elution of high DAR numbers, so the very hydrophobic parts of our ADC sample. So what was used here is an ADC mimic, which contains DAR 0, DAR 2, 4, 6, and 8. And what we see is that we can actually separate things that we cannot separate on any of the other columns. So we can say we have a superior separation performance, and I'm glad it's not only Tozo who thinks it's a very good column. We tested it, or let it test from a couple of beta testers. One of them was working in a company regularly analyzing ADCs, and she said it worked very well. Resolution was higher than with the one that we are currently using. So I'm glad also our customers see the benefit in this column. One thing I also pointed out already was that we had a look at the also injection to injection repeatability of that column. So we tested it with two different molecule types. One was an IgG, which I would personally regard as the easier sample because it's less hydrophobic. And we saw that over 500 injections, the chromatogram is actually very comparable to the very first injection. The second sample we looked at was an ADC, and this I would regard as more complicated because more hydrophobic, and with those we sometimes have more issues, especially if we have a lot of injections. It might be necessary to do a long cleaning step in between, for instance, to get back to the original performance of the column. But here what we see is that over those 200 injections, we really have a very comparable shape of the chromatograms, meaning for at least 200 injections. We did not test more. It might be it goes up to 500 as well. We see a good repeatability. Last slide on this new column, and then we come to another modality of chromatography. I would also like to point out that this column can be used at a higher flow rate, and this means we can drastically reduce the time that it takes to analyze the drug-antibody ratio in a sample. So if we compare a 10 centimeter column versus a 3.5 centimeter column, and again the 3.5 run at a flow rate of 2 milliliters per minute, what we can see is that on the one hand we reduce the analysis time from roughly 19 minutes down to 9 minutes. And secondly, if we calculate the drug-antibody ratio based on these chromatograms, we see that we end up with more or less the same number. Meaning, for this sample, it works perfectly to run the separation just at 7 minutes as compared to, let's say, 18 minutes we would need for the conventional method. Now, I would like to switch gears a little and look into size exclusion chromatography. Size exclusion chromatography, as the name says, separates by size. And what we will find in an ADC sample is, of course, our ADC. We may also find aggregates thereof. But we may also find molecules that are smaller than our ADC. For instance, the free linker or the free payload or the free payload plus linker. And all of this has an impact on the safety of the therapeutic sample we are looking at. So as I said, size exclusion chromatography separates by size. So basically what we have is a column with a porous material. Larger molecules, they cannot enter the pores in the material, so they will straight go down to the column and elute early. If we have smaller molecules, they will enter each and every pore of this column, and that of course takes longer. They will allude later, and this leads to the separation by size that we see in size exclusion separations. Now, the first thing I would like to look into is the aggregate analysis, which is still rather conventional, I would say. And what was done here is a ADC was employed. It was exposed to different kinds of stress, for instance, light or oxidation or thermal stress, in order to get more aggregates and to monitor them by size exclusion chromatography. The column that was used for this was a UPSW 3000 column, so one of our columns. And what we see is that the control without any stress already has a small portion of a dimer that can be bottom line separated from the monomer using the method outlined here. Now, if we look at the different stresses or the samples that are exposed to stress, we will find that the amount of dimers and also of higher molecular weight impurities increases once the ADC has been exposed to stress. For the separation, we can conclude that we have bottom line separation between dimer and anything that is between the monomer and anything that is dimer or higher molecular weight impurity in all of these samples. Now, as I said initially, we may not only have impurities that are larger than our monomeric ADC, we may also come across molecules that are smaller, for instance, the linker or the drug. However, the separation of those molecules is often not as easy on size exclusion columns. Reason being, we may come across interactions with the stationary phase because the linker and or the drug are rather hydrophobic. The payloads are often lipophilic, which may increase the interactions with the column. And for size exclusion chromatography, we need to think back of how it worked. We don't want any interaction. We just want diffusion in and out of the pores. Now, here we see an separation of a lipophilic payload. And if we look into how it's separated, if we don't use any acetonitrile, we see it's not really separated at all. It comes off the column at one point, but also rather late. And the peak shape is far away from symmetric or sharp. However, if we add acetonitrile to our organic to our mobile phase, then we will see that the elution takes place at earlier time points, and we will get sharper peaks. This is because this addition of acetonitrile or an organic modifier reduces the hydrophobic interactions between this payload and the stationary phase. Okay, cool. We are solved now. We can get the lipophilic payload of the column. But what we should still think about is we also have a protein part if we have a complete ADC sample. And typically what we don't want is to expose the protein to too much organic solvent, because it may lead to denaturation of that protein part. So this paper actually developed a very clever method in order to have both separation of the ADC and of the linker and the payload. The way they did it is they initially used a mobile phase with just a very low amount of organic solvent, and this is where very likely our proteinaceous molecule is separated in. Because again, thinking back how size exclusion works, we don't have interaction. We should not have any interaction between our molecule and the stationary phase. And we just have diffusion in and out of the pores. So the protein will probably only be exposed to this low organic solvent throughout the run. Now, what we said before, we don't want any interaction between the analyte and the stationary phase. Is not as true, unfortunately, for our payload or linker. This is what we have seen before. So in order to get rid of those interactions that we don't want, we, in the next step, increase the acetonitrile content of the mobile phase. So we have a step gradient going up to 20% of acetonitrile and then going down again. And this leads to the fact that we can achieve the illusion of payload and linker in nice peak shapes and also in a reasonable time. If we think back of the slide before, the payload came down just at like roughly 13 minutes if we don't add any acetonitrile. With the step gradient, we get back to a very nice chromatogram which separates ADC, linker, and payload for, in this case, two different ADCs. With that, I would like to come to the last part of the presentation, and this looks at affinity chromatography. This is probably not something we think of if we first think of analyzing ADCs. However, if we think of how ADCs work, we will find that, of course, the most important mechanism is we have a chemotherapeutic drug that is internalized into a specific cell and then leads to cell death. However, we may also have secondary interaction, mode of actions, and one of them is called antibody-dependent cell cytotoxicity. This basically works by the Fc part of this antibody, or antibody-drug-conjugate, I need to say in this case, and the Fc part can interact with natural killer cells, and if the interaction takes place, the natural killer cell will release granzyme and perforin, which again leads to cell death in our cancer cell. So this is a secondary mode of action of the ADC, and this is something that we actually copied onto an HPLC column. So what we have here is the Fc receptor, which is exposed on this natural killer cells and is supposed to interact with Fc parts of the ADC or antibodies. And this is what we put onto the stationary phase. Now, if we inject antibodies or ADCs onto the stationary phase, they will bind to this receptor and they will be separated using a pH gradient which is lower by affinity to this specific receptor. Now, this affinity to the receptor is directly linked to how good the molecule acts by antibody-dependent cell cytotoxicity. So how good this mechanism of killing the cell basically works. Now, we use this specific column with that receptor where the chromatogram that we get directly relates to ADCC for different antibodies and antibody-drug conjugates. This is what we see here. This is the first two lines, just rituximab without conjugation and trastuzumab without conjugation. And what we typically see is that we have a 3-peak chromatogram meaning low affinity to the receptor, and high affinity and mid affinity to the receptor. Again, correlating to not so good cell killing activity by the mechanism and very good cell killing activity by ADCC. Now, if we look at conjugated molecules of exactly those maps, we will see that we do have an impact. Not so much on rituximab, for instance. There we see the peaks get a little broader. We have more like a double peak for each of them. But still, we can say we do have some kind of ADCC that is happening here. However, if we look at trastuzumab, if that one is linked or conjugated via this linker to a chemotherapeutic drug, what we see is that most of our molecules elute very early, meaning they don't have affinity to the specific receptor, meaning it hardly has any activity by ADCC, which again is the secondary mode of action of the ADC. With that, I would like to come to a conclusion again, or already I need to say. So what we saw is that hydrophobic interaction chromatography, which is a conventional method to analyze drug-antibody ratios by separating by overall hydrophobicity of the molecules. There we meanwhile find improved stationary phases that allow for very fast analysis and also for better separation performance. For size exclusion chromatography, apart from the standard methodology which analyzes aggregates in our ADC sample, we found a method that can not only look into the ADC and the aggregates, but also the presence of a free linker and a free payload. Last but not least, something very specific, if we want to look at secondary modes of action, one possibility is to use FcR3a affinity chromatography, which contributes to the overall action of the ADC efficacy by an extra ADCC. It's a little confusing, ADC and ADCC, I need to be fair. But still, this is a mechanism that contributes to the overall efficacy of cell killing, basically in the tumor cells or whatever cell is targeted. And one way to use is affinity chromatography using the Fc gamma 3a receptor column. And with that, I thank you for your attention. I hope you heard something new. And if there are questions, please take the opportunity now to ask them, or take the opportunity later to contact me using this email address, or of course your contact at Avantor. I'm looking forward to your questions.
Thank you, Andrea. Thank you very much. So, as usual, it was really interesting, complete, professional. So, thank you for that. Yes, we have received some questions, Andrea, and if you want, I would like to ask you. So,
Sure.
the first question coming from Germany: which detection wavelengths do you typically use for ADCs?
Yeah, there are several possibilities actually. So one typical thing is you could go for 280 nanometers, so just like for normal protein measurements, which basically looks at or detects the aromatic side chains in your protein. However, because like for normal protein, you have one big peak and several smaller ones, but here you typically have them separated. So I would rather recommend to use 215 or 220 nanometers because that looks at the peptide bond rather, and is more sensitive as compared to the 280. nanometers.
Okay. Another interesting question. How can a IC method be optimized?
Okay, that is actually a question that I would need another webinar for, but in a nutshell, you have several things that you can use to optimize. So you can start with how shallow the gradient is, for instance. You can look at the salt concentration that you're starting with and that you end with. You can play around with the pH. You can use organic solvents, not use them at all, or at different concentrations. All of this has an impact on how good the separation will be at the end. And yeah, you have a lot of things to try if it doesn't work at the very first separation. Why?
Okay. Another question concerning mass spectrometry. Is any of the presented methods compatible with mass spectrometry?
Good question, and not too easy to answer actually, because I mean, all of the methods I just showed you used salt in their mobile phases, which is per se not good compatible with mass spectrometry. However, at least for FCR affinity chromatography and for size exclusion chromatography, it works, and there are publications out that show that mass spectrometry can be used together with those methodologies by just switching to a volatile buffer. So that is possible. However, for hydrophobic interaction chromatography, there are also 1, 2, 3 publications out that show compatibility. However, it is much more tricky to get hydrophobic interaction chromatography together with mass spectrometry, and it's not something that is regularly used, I would say.
Okay. Another question concerning lifetime. What is the lifetime of the affinity column?
Good question. As always, it depends.
Not so easy.
I mean, the column also works with rather dirty samples, and if we use something which has a lot of impurities in there, I would say you can be happy with 300, 400 injections you can place onto the column. However, if you have very clean samples, we also did 600 to 800 injections on one and the same column already. Successfully, I need to say.
Another question concerning the IC column. Did you test that new IC column with other molecules as well?
Yes, sure. I mean, what I showed were like the mimic, the etc.s, which is in the ADC, and I also showed, I guess, an IgG, so a normal map without conjugation. However, we also tried it with different other proteins. We also ran plasmids on it already, so it can be very versatile in what you want to use it for, actually.
Okay. So I would like to go again on the question box. So no additional questions. So I think that we can conclude. So thank you very much, Andrea, again for this very interesting presentation. I would like to tell a big thank you also to Florence for the organization of this webinar. Concerning the brochures, you can immediately go on the materials part on the banner, and you can download some application notes and brochures linked to this webinar. You can also, of course, go to our Avantor website to find many brochures on the literature part and you can write chromatography consumables, and after Tozo Bioscience, and you will find many brochures and application notes. So yeah, feel free to download these great tools and support. Tomorrow you will receive the recording. So as I said before, I think that it could be interesting for you to use this recording to share with your colleagues. In some minutes, you will receive a survey. Feel free to give us your feedback, because it's really important for us to know your opinion concerning this webinar, but also some suggestion for the future, because it's really essential for us to receive such a feedback. And again, another point is that in 1 week, you will receive a follow-up mail with many additional information like brochures, application notes, and also some links to our website. So I think that we can conclude, we can close the session. I will check again the question box, no questions. So again, thank you very much, Andrea, for this great webinar. And yeah, I would like to wish you a great evening. And I'm sure that this webinar will be really helpful for the future, for your method development and future methods. Thank you very much. And I would like to wish you a great evening. Thank you. Bye-bye.